Hello, my name is Srivasini Prasanna, and for the Science Fair project, I am presenting my experiment and case study, the effects of Pleurotus ostreatus, oyster mushrooms, on nutrition-deficient soil. So, to gain clarity, what is Pleurotus ostreatus? Pleurotus ostreatus is commonly known as the oyster mushroom, and these mushrooms are a part of a larger family termed as fungi. It drew my attention when I found mushrooms growing next to healthy vegetable plants, and my curiosity led me to investigate about mushrooms. Furthermore, the studies made me wonder if growing oyster mushrooms increases the fertility, to be specific, the nitrogen, phosphorus, and potassium levels of the nutrition-deficient soil. I will also be mentioning NPK. NPK is the short form of nitrogen, phosphorus, and potassium. In my research, I found that many types of fungi have been known to increase soil fertility. I also found that NPK nutrition is vital for the growth of plants. So I created a home lab experiment. It consisted of 1 bag of oyster mushroom mycelium, which would eventually grow into oyster mushrooms and would be the independent variable. I bought NPK soil testers, which would test the NPK levels of the soil. These levels are the dependent variable of the experiment. I would also need 4 cups of nutrition-deficient soil, water, and hay, which I would place into a tank to house the oyster mushrooms. I would make sure that the type of soil, amount of soil, and brand of soil testers would stay consistent throughout the experiment. Once I gathered all my materials to conduct the experiment, I started off my experiment by testing the nutrition-deficient soil and recording my results. Then, I set up my growth chamber by filling a tank with soil and placing a large layer of hay just thick enough to cover the soil. I placed the mycelium on top of the hay and soil base. I then activated the mycelium. During a span of 7 weeks, I observed the oyster mushrooms grow. I would harvest some matured mushrooms and leave others to decompose. After 7 weeks, I tested the soil and collected the data. I found that the NPK levels of the soil had increased or maintained levels according to these photos. I plotted my readings in a graph and chart format. In this chart, you can see that before oyster mushroom growth in the soil, nitrogen levels were depleted, but after growth, levels were sufficient. Phosphorus levels increased from adequate to surplus over the period of experimentation. Potassium levels had stayed the same throughout the experiment. From the data, I concluded by stating that Pleurotus ostreatus, commonly known as oyster mushrooms, do increase or maintain nitrogen, phosphorus, and potassium levels of the nutrition-deficient soil. In slide 2, I've posted photos depicting the various growth phases of the oyster mushrooms. I have also posted additional photos of the NPK testers compared. In the pictures that represent the phases of the mushrooms, you can see how the mushrooms grew. In phase 1, the mushrooms are small white stubs. Phase 2 shows the mushrooms have grown taller and have convex-shaped tops. In phase 3, the mushrooms have transitioned into bigger, matured mushrooms with cup-shaped tops. Phase 4 is when the mushrooms are either harvested or left to decompose. In this picture, this particular oyster mushroom was very large, about 1 foot tall. Looking at the pictures that represent the soil testers, we can see the change of colors in testers. Each tester has 5 levels, which are 0, depleted, 1, deficient, 2, adequate, 3. Sufficient, and 4. Surplus. Consider the picture of the pink soil tester, which is nitrogen. Before experimentation, there is almost no color and it looks clear, but afterwards, it was an opaque pink. Phosphorus testers show that the blue has changed from a light blue into a deeper, more saturated blue. The potassium color had stayed the same and hadn't changed. These images are a visual representation of the nutrition levels that help me conclude that the introduction and growth of Pleurotus ostreatus oyster mushrooms to a nutrition-deficient soil had enhanced or maintained nitrogen, phosphorus, and potassium levels in the soil. This third slide represents my abstract. I hope you enjoyed my presentation as much as I enjoyed sharing my research and experimentation with you. Thank you.